So what became clear was we had these bacteriophages, they're different morphotypes, and when they're added to a bacterial host, they can reproduce. And a great deal of work went into understanding what the principles of that reproduction are. And although, again, there are variations on the theme, I'd just like to go over the core elements of the theme as to what's happening during that viral reproduction. So we can begin with this first step here, where here is our bacteriophage particle, and it's going to infect this bacterial cell, showing it's the outside of the cell wall here and the genome. It's important to note that in this first step, which is called adsorption, that there is a specific recognition, a specific interaction that occurs between the structures on the tip of the tail, irrespective of what kind of tail it is, between the tip of the tail and the outside of the cell. In the next step of the process, the genetic material, as I said, typically double-stranded or frequently double-stranded DNA, is injected from the head through the lumen of the tube into the inside of the cell. The rest of the particle, the proteinaceous components, the head and the physical tail, remain on the outside of the cell. They do not enter the cell. And so this particle remains there, and it's only the nucleic acid that penetrates into the inside of the cell. In a typical cycle of lytic bacterial growth, what happens is that the virus uses the machinery, the molecular machinery within inside the cell, to reprogram the cell so that it makes lots of more virus, and ultimately results in the death of the cell and the release of the new viral particles. And so there is a well-defined program of gene expression and protein function that occurs after the DNA has entered the cell. The phage genes are expressed in order to make proteins. These proteins are often expressed in different temporal patterns, so you may have early protein synthesis, and what's shown here is late protein synthesis. There may be many intermediate steps of protein synthesis that some viruses use. Most importantly, there's really two key things that have to happen to reproduce the virus. You have to make a lot more viral DNA, and then you have to make a lot more particles. And so the process involves making the machinery that's needed to assist the cell in making lots more copies of the viral genome. The proteins have to be made that will assemble to form the viral structures themselves, the heads and the tails. There's a well-defined process of assembly in which the heads and tails are usually formed as separate structures. The viral DNA is packaged into the head, and then the tail is added. The final step that has to happen is what is referred to as lysis, where the integrity of the membrane and the wall is interrupted, it is broken, and the baby bacteriophages, the progeny phages, get to exit the remains of the cell and to go on and do the same thing all over again. The number of bacteriophage particles that are generated from a single infection is referred to as the burst size. The burst size varies from phage to phage. It is often between 50 and to 100. Can be more, can be less, but those are typical numbers. And so that burst gives you 50 to 100 progeny from a single infectious cycle, from a single infectious agent going through this replicative process. All, or the vast majority of bacteriophages, can do lytic growth. The process that I just described is lytic reproduction, making more of themselves. But many bacteriophages, and some might argue perhaps most, can do other things as well. And there are a large set of bacteriophages which can establish a state that is known as lysogeny. And phages that are able to form lysogens, as well as going through lytic growth, are referred to as temperate bacteriophages. And I'm going to take a couple of minutes and just describe how the lysogenic state and the establishment of the lysogenic cycle differs from lytic growth. One way to think about it is that in this infectious process here, where the bacteriophage is going to infect these cells, that a choice is made, a decision is made, and it's a developmental decision. It's a choice as to which of the two possible outcomes will occur. So one outcome is lytic growth, which is exactly what I just told you about, where the viruses reproduce, you get cell lysis, so the cells are dead, and you get lots more progeny bacteriophages. When lysogeny is established, the cells do not die, the viruses do not reproduce, at least there's no generation of new particles. And in fact, what happens is the bacteriophage DNA, instead of going through the reproductive cycle, establishes what is, in essence, a parasitic state, where the phage DNA is co-propagated with the host stably over large numbers of generations. So I'll explain that a little bit further, but I want to emphasize that these so-called lysogens, these lysogens where now the bacteria are carrying with them this form of the viral DNA, while stable, is not terminal, by which I mean that there is a process where these lysogens can be induced into the process of lytic growth. And this induction can happen spontaneously, low frequency, maybe 1 in 10,000 or 1 in 100,000 cells per generation. But often that frequency can be stimulated by treatments which damage the cellular DNA, UV, mitomycin C, many other similar types of treatments. I should emphasize that whilst many different bacteriophages of many different hosts have been shown to undergo this UV or mitomycin C induction, it doesn't appear to be true for all bacteriophages, all temperate phages, and all lysogens. So, so this diagram just superimposes this form of lysogenic growth alongside this lytic growth. And so what I told you was that when the bacteriophage DNA infects the cell, instead of going through this reproductive state, the viral DNA becomes established as what is referred to as a prophage. And that prophage is often, but not always, integrated, so it becomes part of the bacterial chromosome, and the DNA replicates just as though they were genes on the bacterial chromosome using the host machinery, and all of the viral genes that are needed to do this lytic process are switched off. That process is quite well understood in some cases, and I won't go into it in detail, but the concept is that there is a transcriptional repressor which is synthesized in these lysogens, binds to operator sites on the prophage DNA, and represses and shuts down this program of lytic reproduction. And so as these cells grow and divide on and on, the prophage DNA becomes replicated at the same time. And as I said, this is a stable, genetically stable state, so the prophage can reproduce for many, many generations until such an event such as spontaneous